Every human brain begins as a tangle of chaotic tissue. To wire a functional nervous system, millions of axons have to navigate this dense, microscopic wilderness, seeking out specific targets to form the circuits that process thought, movement, and memory. This routing process happens at a staggering scale and speed. Billions of connections are established simultaneously, forming a network so dense it defies manual mapping. For decades, the primary assumption in neuroscience has been that this immense complexity must be governed by a deterministic blueprint. The logic suggests that the genome provides a strict schematic, using precise molecular markers to guide every single axon to its preordained destination. This blueprint model directs the focus of modern research toward finding a static map. However, if the brain's development doesn't actually follow a fixed schematic, we risk searching for a set of instructions that the genome never actually wrote. Evidence suggests an alternative. Brain wiring operates as an irreducible, unfolding evolutionary algorithm. It is a dynamic process of self-organization rather than the execution of a pre-planned map. This distinction changes how we approach intelligence. If we assume a blueprint where there is actually an algorithm, we will design genetic screens and artificial neural networks that miss the very principles that allow complex biological systems to organize themselves. To understand biological development as an algorithm, we can look at the theoretical baseline for complex systems, established by Stephen Wolfram through his work with cellular automata. This animated grid visualizes Rule 110. It shows how incredibly simple, deterministic local rules for black and white pixels generate a highly complex, asymmetrical pattern as the computation cascades downward. This illustrates a mathematical concept known as computational irreducibility. Computational irreducibility means there's no mathematical shortcut to finding the end state. You cannot predict the final pattern by looking at the starting conditions. The only way to know the outcome is to actually run the algorithm, step by step, to its conclusion. This constraint applies directly to biological growth and the wiring of the nervous system. In a developing embryo, the governing rules are not static. Genetic expression changes with every iteration of growth, shifting the algorithm's parameters mid-computation. Furthermore, biological development operates in a physical space subject to stochastic noise, random chemical and mechanical disruptions forcing the system to adapt. Evolution accounts for this unpredictability through brute force. It runs the full, noisy, irreducible program to whatever end it reaches, simultaneously across millions of different individuals in a population. Selection happens after the growth program is complete. Evolution chooses based on the final outcome, whether the organism survives, without needing to see or predict the intermediate molecular steps that built the brain. Because development is computationally irreducible, looking for a direct mapping between a specific gene and a final behavioral outcome is mathematically incomplete. The genome encodes the process, not the result. Algorithms are complex and non-intuitive. Science often gravitates toward clean, solvable mechanisms that offer a clear line of cause and effect. This preference for elegance is why Roger Sperry and his chemoaffinity hypothesis became the cornerstone of developmental neurobiology. This diagram illustrates the mechanics of chemoaffinity. It proposes that incoming axons carry specific chemical markers that seek out complementary surface proteins on the target tissue. When these markers align, the connection is fixed. This theory suggested a synaptic address code, where every neuron has a distinct molecular signature directing it to its exact destination. The scientific community adopted this model because it provided a direct, testable link between a single molecule and a specific wiring outcome. It turned a messy algorithmic problem into a manageable molecular one. We see the legacy of this in modern genetic screens. When searching for wiring defects, researchers often filter their data to highlight specific classes of molecules, like cell surface receptors, while discarding others. A mutation in a receptor is prioritized because it fits the model of a recognition molecule. It offers a clear path for further study. Conversely, mutations in mundane metabolic enzymes or housekeeping genes are often sidelined, precisely because they don't fit the narrative of a targeted blueprint. Sperry's model dominated the field not because it explained the entire system, but because it provided a deterministic target that was accessible to molecular biology. The strict blueprint model breaks down when confronted with experimental anomalies, specifically experiments where exactly half the target tissue in a visual system was surgically removed. Under a strict chemoaffinity model, destroying half the target markers should leave the axons matched to those markers permanently stranded, unable to dock anywhere. But that isn't what happens. The incoming axons reorganize. They dynamically form a fully compressed functional map within the remaining half of the tissue. To explain this, researcher Michael Gaze proposed an alternative: relative positioning, or systems matching. In relative positioning, axons don't look for a unique address. They navigate by reading intersecting chemical gradients, finding their destination by calculating their position based on the shifting ratios of these spatial values. Gaze's theory describes a self-organizing process. It relies on continuous feedback and relative rules, rather than a static, pre-planned map. If Gaze correctly identified the principle driving this behavior, why is his work largely secondary to Sperry's in the modern curriculum? The reason is that relative positioning cannot be traced back to a single address molecule. Algorithms are built from multiple, mundane molecular functions acting in sequence over time. They are not controlled by a discrete molecular driver that is easily isolated in a genetic screen. Because of this complexity, the field has often reacted by diluting the definition of recognition molecules, stretching the term to keep new discoveries within the familiar framework of the chemoaffinity paradigm. By expanding the old blueprint metaphor to include every new discovery, the field essentially marginalized the algorithmic hypothesis in favor of a simpler, more intuitive narrative. Advancing our understanding of neural circuitry requires moving beyond the search for deterministic blueprints. We must accept that brain development is a permissive process, one that relies on an algorithmically unfolding systems match rather than a hard-coded schematic. This biological reality reflects how we approach the architecture of artificial neural networks. We do not build an ANN by hard-coding every individual connection. Instead, the network organizes itself by adjusting weights through a learning algorithm. We provide the rules for adaptation and allow the optimal network to self-assemble over time through trial and error. The final map is the result of the process, not the starting point. In both biology and AI, demanding a blueprint can lead us to ignore how the system actually works. We have to see the data first, and only then build the theory. Whether we are sequencing genes or writing code, if we look for a predetermined map, we will fail to understand the irreducible algorithms that actually generate intelligence.